Great. Well, I'm going to kick this off. Thank you everyone so much for joining our webinar on visualizing and annotating single-cell data with Genome. My name is Hannah Schuyler. I am a Director of Business Development here at Genomics, and we also have our in-house single-cell expert, Nora Kearns, joining us as well. So thank you so much, and excited to be talking through some of these capabilities with you today. So just a few quick things before we get started. This webinar is being recorded and will be sent out to all attendees. So thank you for joining. And then please enter any questions that you have for the presenters in the chat box, and they will be addressed at the end during the live Q&A session. So definitely want to make sure that we get those addressed and type those in the question box for us. So what will we be discussing today? We have a few things that we'll be covering. First, I'll go over an overview of Genomics, so who we are, where we came from, and how we're helping a variety of our different clientele analyzing genomic data. Then we will talk through how you can actually visualize and annotate your single-cell data with our guided workflows, allowing you to go from raw data to processed without having to use code. We'll leave some time for live Q&A at the end to go through any questions that you guys have. And then lastly, we'll be talking through next steps on those that are interested in testing and piloting the software as well. So just to give you guys a little bit of context in terms of who we are a spin-off company from IBM Research Center in Almaden, California. And the software was originally built as an internal need for pet and food safety. And the team quickly saw that there was a need for building out these pipelines internally, especially for those that did not know code. And so that's how Genome was born. And now we work with a variety of different clientele, including pharmaceutical clients, academic clients, biotech clients, to help them with their data analysis, whether that's using some of our pre-built workflows that we're going to talk through today, access to curated data sets from some of our partners, as well as bioinformatics consulting services as well. So the goal here at Almaden Genomics really is to help streamline the collaboration between the data scientist and the biologist. It's a very complex space, as I'm sure many of you know. The biologist has a goal of analyzing their own data, whether it's in-house sequencing data or public data, and their biggest priority is really creating visualizations and making sense of that data. And it's possible that they may not know code. So having to build a pipeline might be a little bit scary for them. And so they often are relying on bioinformaticians to do this analysis for them. This can be a time-consuming process. There's not necessarily a common language between both parties, and it's not necessarily an easy process. And then, of course, on the other side of the coin, we have the bioinformatician. Their main priority is building pipelines, workflows, leveraging open source tools, and really being tasked with helping the biologist analyze their data. And so our goal here really is to help kind of streamline that process and bring the collaboration between both of those two teams. And so, of course, today's webinar is focused on single cell. And I think it's important we touch on why this type of analysis is so important. And before I jump into some of the reasons why single cell is so important, I'd like to host a poll and see how many of you are actually working with single cell data right now. So in just a second, you should see a poll pop up. So are you working with single cell data now or planning to do so in the near future? Go ahead and answer if you see that come up on your screen. So yes, I am working with single cell data now, not yet, but I'm planning to in the near future, or no, I'm curious to learn more. Awesome. It looks like we kind of have a combination of those that are working with single cell data and some that are interested to learn more. So thank you so much. And I think this is a perfect transition as to kind of why single cell is so important. I'm sure many of you have worked with bulk RNA-seq data in the past. And what's so powerful about single cell analysis is that you're able to uncover insights at the cellular level versus an average of what is happening in all of the cells, which is typical of bulk RNA-seq, right? And so with single cell analysis, you have the ability to analyze the gene expression profiles of individual cells, which can also reveal cellular heterogeneity and rare cell populations that might be masked when you're looking at your bulk RNA-seq data. And then lastly, single-cell analysis have really been critical in furthering scientific discoveries. Some of those may include how to predict how a disease may evolve over time, uncovering the mechanism of action for different drugs, or even discovering new cell types. So while single-cell sequencing is very transformative with a lot of potentials, there's several challenges that we hear from researchers that they face when it comes to analyzing these complex data sets. And the first being is that these single-cell data sets are typically quite large, making storage and processing very computationally intensive. And so this means that there's longer computation times for the researchers, as well as potentially higher costs in actually analyzing this data. There's also a lack of standardization for data analysis as it relates to single cell. Over the years, we've seen so many different platforms, open source tools, technologies that have been deployed to process single cell data, which of course complicates any sort of cross-study comparisons, data integration, or standardization as you're looking to process these different types of data sets. And then lastly, being able to accurately identify and annotate cell types is a very time-consuming process, which I'm sure if you are working with single-cell data, you're very familiar that this is sometimes a tedious process. And so clustering of the cells really doesn't mean anything if you aren't able to identify and annotate what those clusters are. So there's typically two different approaches. Researchers either must do this manually to identify cell types, meaning they would have to know the marker genes associated with their cell types, or use some sort of automation. The catch with the automation, of course, is that you need to have some sort of training data set or reference data set. And as you can imagine, both of these types, or I guess approaches, have their own challenges. You either are unsure of what you're looking for in the cell types, or you lack a strong reference. And so it's a common challenge that researchers face quite frequently. And so the goal here really with our software Genome is to help reduce some of those complexities for researchers with our pre-built workflows that can be used by biologists, data scientists, and bioinformaticians. There's many different ways you can actually bring in different types of single-cell data. So one of those options is the ability to pull in public data from our SRA integration and run it through our SRA tool. So accessing any of those public data sets can be processed on our software. You can also process in-house sequencing data files. So if you're pulling that from a local server or an S3 bucket, and then you also have the option to really process that data at any point in the journey. So whether that's starting with a FASTQ file, an HDF5 file, or even a filtered gene barcode matrix file. And then we're also working with data partners to bring in those curated data sets so you can process, analyze, and visualize those data sets on our software as well. And the goal is that you would start with a pre-built workflow, right? So we're using Seurat or Star Solo, go from the raw data files to those meaningful visualizations. But one of the benefits about the software is that you can actually do a variety of different custom modifications to the workflow. So you have the option to modify parameters. You could do batch correction, adjust normalization, evaluate cell cycle genes, and even filter out cells. And then with this guided workflow that Nora's going to talk us through in just a bit, you also can perform cell type annotation with access to over 28 different reference data sets. So this, again, helps kind of cut down on that time to actually annotating the cell clusters for you. And then lastly, we also produce interactive reports and visualization. So you get UMAPs, violin plots, cell counts, top genes. All of this can be imported into Jupyter notebooks as well to take kind of one extra layer in terms of making it more customized based on what you guys are specifically looking for. And so with that, I would love to pass this over to Nora to give a more formal walkthrough of how you can actually set up a guided workflow on our software and how this will translate into visualizations and interpreting of your single cell data.
Great, thank you, Hannah. Okay, screen sharing, good?
Screen sharing, good.
Awesome. Okay, so Today I'm going to talk to you a bit about the genome software, but really what I want to talk to you about is how I approach the analysis of single-cell data, some interesting insights that we can gain from single-cell data, and honing in on the whole meaning of the webinar, which is visualizing and annotating single-cell data. So just to start, when you enter a genome, this is your project management space. So for each analysis that you are performing, you'd want to create a project. I have already done this cooking show style, so I'm just going to dive into this project we have here. And this is our project management page, so we can, see the cost of our project, manage our project, change metadata, things of that nature, or delete the project. Okay, so when we're running a single cell analysis, the first thing that we have to think about is how we're going to get our data from point A to point B. This is a problem that bioinformaticians experience all the time because bioinformatics data tends to be very large. You might have files that are 10 to 20 gigabytes. And so it can actually be difficult to, download your data and then upload it somewhere else. Those are challenges that bioinformaticians commonly run into. So on g.nome, we have two ways of helping you with that challenge. The first is that we can configure setup to an S3 bucket if you're using S3, so you can import your data directly from that S3 bucket. You can also import files locally from your local machine using a high-speed file transfer tool called Aspera. So either of those are options to help you get your data on platform. I've already imported a data set that we're going to work with today, but once I have my data in the right project space, then I get to start thinking about running my analysis. The genome workspace can kind of be thought of from two perspectives, and it's meant to support two types of scientists. So one is the scientist who has the biological know-how. They know how to set up experiments, prep sequencing libraries, and run that data, but they may not have the coding expertise to perform analysis, with code. And then we also have the bioinformatician who does have a bit more expertise on how to set up these workflows. And this tool or genome is meant to support both of those kinds of scientists. But I first want to talk about the guided workflows because those are our workflows that are really meant to support the biologist. So I'm going to run a guided workflow. We have guided workflows available for both RNA-seq and single-cell, but I'm obviously going to focus on single-cell today. So I will select that workflow. I can label the run however I want to. And then we have to select our input data type. So when you are performing single-cell analysis, you may be starting from two different points. You could be starting with raw sequencing data if you've done the experiment yourself and you want to analyze it. So that would be in FASTQ format. Or you may be starting from the point of a count matrix. So possibly you've gone out and looked through papers, you've found some interesting data hosted on Geo, you want to pull that in and try to reproduce their analysis or gain additional insights from someone else's public data. So we have the ability to start from either point. But for today's webinar, we'll talk about FASTQ data. So from my data bucket, I would select my data set. Just takes a second to load. There we go. So I have this PBMC 1K FASTQ data set with all of my files contained within it. Then I'm going to select a sequencing chemistry. So we support 4 different chemistries, 10x 3' V2 and V3, and then 10x 5' paired end and R2 only. Then I need to pair my FASTQs. So this is essentially creating a sample sheet. And the important functionality about this is that it ultimately is what parallelizes the processing of your data. So you might have 5, 6, 10 samples, and you want to make sure that those samples are processed in parallel all at the same time, rather than painstakingly going through and processing each sample individually. So this tool is essentially taking care of that for you. And I'm going to leave the sample names as default, but you also have the ability to go in and change those to whatever you want them to be. So once we've paired our FASTQs, we need to create a metadata file to associate the samples with important biological characteristics. So you may have a sample metadata file that you can import, or you can manually create a metadata file. So I'm going to just have an experimental group and a control group, and then associate each sample with its correct condition. Then I want to select my reference genome. On genome, we supply the GRCH38, so human reference genome, and the MM10 mouse genome. These are specifically configured by Cell Ranger for Cell Ranger data. But if you are working with a non-human or mouse organism, let's say you're working with rat or zebrafish, you can also supply a genome FASTA file and a GTF file, and the workflow will generate your reference genome for you. Okay, I want to take a quick pause here and talk about the steps that we've been through up to this point. So we have imported our FASTQ files, created a metadata file, and selected a genome. All of those steps are important for running the first part of single-cell analysis, which is QC and alignment. QC means evaluating the quality of our sequencing data. So actually, how good was the imaging of your flow cells? How good is your data? And then alignment is aligning the RNA that was sequenced that's in your FASTQ files against a reference genome and counting the number of RNA transcripts observed from each gene. So it's generating the counts data. So everything up to this step has helped us generate the counts data, or set us up to generate the counts data. Now we have to start thinking about the actual counts data, and the first step in doing that is filtering parameters. So there are a few filtering parameters to think about. There's maximum RNA features per cell, minimum RNA features per cell, and max mitochondrial DNA. So maximum RNA features per cell can help us eliminate doublets, which are occurrences of multiple cells kind of stuck to each other that are observed as a single cell. So we're getting way, way, way more counts than we should be getting if it was from a single cell. So we can set a maximum for that. The default is 10,000, as well as minimum RNA features per cell. If a cell is barely expressing anything, we probably want to exclude it. A good default value for that is 200. And then lastly, max mitochondrial DNA. So you want to set a maximum mitochondrial DNA percentage because mitochondrial or high quantities of mitochondrial DNA can be indicative of cell damage and cell stress. So we want to get rid of cells that have very high counts of that. Generally, a good range to go by is 5 to 20%. PBMCs, which is the type of data in this example, you can typically go a bit higher, so around 15 to 20%. So that's the default value that we have set there. Then you want to just decide if you want to evaluate cell cycle genes. So at the end of a single cell analysis, you get these beautiful clusters, or hopefully beautiful clusters, and ideally you want those cells to cluster together based on type, but there are lots of different reasons why cells may cluster together. Ultimately, clustering is a sign of similarity between cells. So there are other reasons why cells may be similar, and one of them is that there are cells in the same stage of the cell cycle. So because they're expressing the same set of cell cycle genes, they are similar to each other and they may cluster together, but we don't really want that to happen. So we can choose to evaluate and regress out the effect of cell cycle genes on our data. By default, the human cell cycle genes are preloaded in Seurat, but you can also choose to supply your own input S-phase genes and G2M phase genes. Okay, then we want to think about, if we have multiple samples, do we want to integrate and correct for batch effects in those samples? So batch effect correction is correcting for technical covariation in the data that is the result of technical setup in your sample prep. So for example, you processed or prepped your first sample on Monday at 10 a.m. and then you processed another sample on Tuesday at 10 a.m. Or two different people prepared those individual samples. That can create unwanted variation in the data that's solely the result of that technical reason, but it's not indicative of underlying biological difference in the data. So we want to prevent that from creating unwanted differences. So let's say we do want to perform integration and batch effect correction. Then we have to decide what algorithm do we want to use. Seurat by default chooses or uses CCA. And CCA is a really good algorithm to use when you expect to see conserved cell types across your conditions, so the same cell types across your condition, but there may be significant shifts in expression that are the result of your experimental conditions or disease states. Like you might have 2 different donors with 2 different types of breast cancer. It's also very good when you're integrating across modalities. Another option is to use RPCA, which is for reciprocal PCA. RPCA is great if you have a very large data set because it is less computationally intensive and significantly faster than CCA. And it's also good when you expect to see differences in cell types across your sample. So essentially prevents overcorrection and helps preserve biological variation in the data. So let's go with CCA for these ones because I anticipate seeing the same cell types. And then lastly, we have to think about whether or not we want to perform cell type annotation. On Genome, we support reference-based cell type annotation, which means that we have a data set of already annotated cells, and we are going to compare it to your unannotated cells and try to find similarities in the expression of annotated versus unannotated cells to assign annotations to your cells. So we use a tool called Single-R for that. Single-R examines a reference data set and assigns annotations to your cells based on that. The reference data sets that we have available, there are 28 of them hosted by the CellDex library and the scRNA-seq library. CellDex hosts 7 well-validated data sets that are generally compiled from individual bulk RNA-seq data sets. So they took a single cell type, ran bulk RNA-seq analysis on those cell types to establish an expression profile, and then compiled them. So these are really strong reference data sets, but they are a bit skewed towards immune data. So they're great if you're working with PBMCs, for example. But if you're not, we also have the data sets available from scRNA-seq. These are from individual publications, but they do have more diverse tissue types represented, which is great. And our data sets do span human and mouse. So let's say I just go with the CellDex Monaco immune data, go with that, click next, and then I see a summary of the run that I am about to launch. So I would just click launch. I've already done that and I have a run ready for us to view. I'll click launch on my run, and then it will take me back to this guided workflow page that shows me the runs that I have previously done. So I'm going to go in and view the results of this run that I did yesterday. And so I have all my results divided into individual directories indicating what the results contained in them are. So we'll go into this HTML report directory, and then I would just download the report and open it up. Oh, sorry, over here. So during the single cell workflow, there are multiple steps that happen. There's filtering, there's identifying variable features, running or generating a UMAP, and each of those steps produces visualizations. And essentially what we've done here is we have compiled all of those visualizations into a single report that allows you to download it and share it across your team, for example. So let's dive in to some of these visualizations and talk about what's meaningful about them, what kind of insights we can gain from this data. So the most interesting thing that you get at the end of single cell analysis is your beautiful UMAP, or hopefully beautiful, or a t-SNE plot. So what we show you here are the annotated cell types if you performed cell type annotation, as well as how Seurat identified your clusters. And this can be kind of interesting because Seurat may find further differentiation between specific groups of cells than your annotations would indicate. So it's just kind of 2 perspectives of looking at your t-SNE. We also show you cell counts, so the number of cells observed in each cluster. And this can be interesting if you're comparing 2 different groups. Do I see way more monocytes in one sample than I see in another? As well as the top genes expressed from each cluster. Here we're just seeing the top 10, but you can change it to observe the top 100 or top 50, however you want to do that. And then lastly, this interactive gene visualization. So this is what helps you look at differential expression of genes across your clusters. So for example, here I see this CD6 gene is really strongly expressed in this group of, let's see what these are. These are T cells. So I see that that's really strongly expressed in this group of cells. If your clusters are not what you expected them to be, then you can kind of take a look at your QC and see, maybe my data isn't super high quality, or maybe my filtering wasn't great. How can I adjust things to get closer to what I would expect to see? So we show you your QC results. You see here the distribution of total transcript counts in each cell, the distribution of genes observed in each cell in the center here, and then the distribution of mitochondrial DNA. We also show, this is essentially the same data in a different format. So, RNA transcripts on the x-axis here versus mitochondrial DNA, or number of unique genes versus transcript counts. This perspective of the data can be really interesting because it can help you identify patterns of quality in your data. So this data actually looks pretty good. You generally want to see kind of a nice tight line here. But let's say that you had, two different lines that were kind of shooting out on this plot. That may show you that, oh, there's some kind of technical thing going on in my data where one group is higher quality than the next. So it just helps you kind of learn more about the quality of your data. We also show a variable feature plot, so the top variably expressed genes within each sample. And then lastly, an annotation QC plot. So you can see there's a lot of annotations here, so lots of things on the x-axis, but basically this delta distribution plot is showing the difference in confidence between the first place annotation assigned to each score and the second place annotation. So you would want the delta to be high because that would mean there's a very clear winner in the annotation assigned to your cell. So on that subject, I want to dive a bit deeper into the subject of annotation. 1 second, I'm trying to move this screen over. There we go. Okay. So when we're performing cell type annotation, we want you to think of running analysis of single cell on genome as being very similar to running an experiment at the bench. So you may get to the point of clustering of your cells and you see that those clusters are not really what you'd expect. They're not clustering well. So there's a lot of things that you can adjust, and that's totally normal and accept. It's not like you are, fudging the data at all. You need to try some different filtering parameters. Maybe the first time you filtered your data wasn't the best. You need to go back and adjust some things. That's very normal in the processing of single cell data. And in fact, many people say that you should expect to spend about 80% of your time in single cell analysis in this space of trying a few different parameters out and getting to the point of what you as a biologist would expect to see in your data. So I'm just going to show you how I annotated the single cell data using 2 different reference data sets. So this is the same t-SNE annotated with the human primary cell atlas and the Monaco immune data set. So let's dive into what we're looking at here. We have this t-SNE, and it's broken out into monocytes, B cells, NK cells, and T cells. How do we know that those annotations were actually good annotations? So this heat map on the right here is how we would evaluate that. So what we're seeing in this plot is that each of these individual little columns, like these stripes, is representing a single cell. And the colored bars across the top are showing the groupings of cells. So this yellow bar is showing the B cells, this blue bar is showing the monocytes. And you can see that the kind of sizes of these chunks of yellow are corresponding to the approximate sizes, number of cells that we're observing on the UMAP. And then down the right-hand side of this heat map are all the possible annotations, essentially. So let's talk specifically about this group of monocytes here. Basically, the algorithm was very confident that these cells were monocytes because in the monocyte label, it's yellow, so it's very high confidence in the annotation. But as we go down, the possible annotations of CD4 positive T cells, CD8 positive T cells, B cells, and NK cells, it's blue. So that meant the algorithm does not think that is any of those cell types. In contrast, the T cells, there's a bit more fuzziness here. So the algorithm was pretty confident that a single cell was a T cell, but it was also uncertain. It might be a CD8 positive T cell as opposed to a CD4 positive T cell. And this is to be expected because within a certain cell type, more genes are, the expression of genes is largely shared. There's just a few genes that are differentiating the two cells from each other. So you'd expect to see that the algorithm has a bit harder time discerning within a specific cell type. In contrast, here is the monoco-immune data set. So this is nice because we get really high resolution or granularity of cell types in the annotation. So we can differentiate between non-switched memory B cells and switched memory B cells. And that's really interesting to see. But as you can imagine, it introduces a lot less or a lot more ambiguity and less confidence in the exact cell type. So within a certain cell type, you can see like this group of cells we're pretty confident is monocytes, but is it a classical monocyte? Is it an intermediate monocyte or a non-classical monocyte? Then it becomes harder to differentiate. But this is an example of the kind of iteration that you can do on genome, and that's something that we want to support. And one specific characteristic that we use to support that is cloning our runs. So we don't want you to ever have to go all the way back to the beginning of your analysis and run it all the way to the end, because cell type annotation is kind of the last step you perform. You can just clone your run and run only this step on, 2 or 3 different data sets until you see what you're kind of expecting to see. The next thing that I want to talk about is batch effect correction and data integration, which is also really important in gaining confident insights into your data. So here's an example of 2 different breast cancer tumors. One of these tumors is HER2 positive and estrogen receptor positive. The other one is just HER2 positive. So we have each individual sample processed without batch effect correction on the left here, and then we have the data batch effect corrected and integrated on the right. If I were looking at these unintegrated samples, I would observe, okay, I have these like 2 large groups of cells across the samples, and they may be really similar, but I'm not sure. But once I run batch effect correction, I can say with confidence, okay, these are the same groups of cells across my 2 samples. So these are really similar, but I am starting to see some interesting differentiation across the 2 samples. For example, this cluster here is largely observed or this cluster of cells here is dominant in the HER2 positive tumor. And, this cluster of cells here is mostly observed in the HER2 positive ER tumor. Once we've done that batch effect correction and integration, we can annotate our cells and start visualizing how different genes are expressed differently in the 2 tumors. So just as an example of this, I've taken the gene SFRP2 and visualized it across the 2 different tumors. And I see that, one, there's higher across the board expression of that gene, but particularly this group of cells here is strongly expressing SFRP2 compared to kind of weak expression in this other tumor. This group of cells is a set of cancer-associated fibroblasts, and this is consistent with what we would expect to see because SFRP2 is a marker gene of CAFs. CAFs are interesting because they can promote tumor aggressiveness. And so this helps us understand, why might this person be responding better to treatment or worse to treatment? Why might this person's tumor be growing more aggressively than another person's tumor? This is the kind of meaningful insight that single-cell data can help us gain. Okay. So that is everything that I wanted to show about the guided workflows and the outputs of single-cell data. Going a step deeper, the guided workflows, the goal there is to, 1, empower biologists to run analysis with autonomy. You don't have to wait on a core to do your analysis for you, or wait on a data scientist who might have 15 different projects going on. You have the power to run that analysis yourself. But we also acknowledge that you're scientists by nature, and you are curious and you want to know exactly what's going on in this kind of analysis, even though the goal of the guided workflow is to eliminate the complexity. But at the end of the day, you might want to see that complexity. You might want the ability to fine-tune and adjust your analysis. So that's what the Canvas view allows you to do. And this is also a nice view for the bioinformatician who wants to know what kind of tools and parameters are being used. So in the Canvas workflow, we have 4 different plug-and-play tools. So QC and alignment, analysis, cell type annotation, and then complete single-cell RNA-seq, which runs all 3 of those in sequence. So to dive in to one in particular, this is the plug-and-play version of the single-cell analysis workflow. So it has the same kind of input parameters that the guided workflow had, such as whether or not you want to evaluate cell cycle genes, or run a UMAP, run a t-SNE, but there is a bit more fine-tuned control here. And if you want to see, you want to have that control and you want to see what's going on under the hood, we can click this sub-workflow button and it will pop us out to the actual workflow that's being run. Okay, and so now we can see the individual tools that are running in sequence for this workflow. So we have a Seurat pre-processing tool, Seurat normalization, Seurat find variable features, Seurat cell scoring. So lots of Seurat. And the whole workflow is powered by Seurat. So if you are a bioinformatician and you're familiar with the Seurat workflow, basically each of these tiles is a graphical representation of a Seurat tool. All of the inputs that are exposed in that tile are the same as the inputs for that Seurat tool. So you have the ability to go in, and I've just done this here. Normally the Seurat find variable features algorithm returns the top 2,000 most variably expressed genes in each sample. Here I've just changed it to 1,000. That's just a random example of how you could go in and change individual parameters in your analysis. So basically by drilling down to this level, you can see exactly what's going on. And also that helps with reporting. So if you are writing a publication and you want to show exactly how you conducted your analysis, this makes it a lot easier to do that. And it also allows you to really understand the underlying workflow. Okay, so that is kind of a wrap on the single cell stuff. The last thing that I wanted to talk about is a common thing that we do as biologists, which is we want to look at someone else's paper and understand it, dissect it, and reproduce the analysis. So that can sometimes be difficult because of the size of the data that you are working with. So let's say someone that has hosted a bunch of FASTQ files on SRA, how are you going to get access to their data, pull it to a place that you can work with, and reproduce their analysis? So a specific tool that we have on Genome to allow you to do that is called SRA import. With SRA import, you will essentially import an SRA file which contains the accession numbers to that data on SRA. So if we view this file, you can see I just have a small data set here with 2 different accession numbers. Then we can go into our SRA import tool and attach that file into the required inputs, and it will create this FASTQ output. We just run the tool, go to our Runs tab, and look at the outputs. So first, or before I show the individual files, I just want to highlight, this is 43 gigabytes of data. This is 355 gigabytes of data. You may not even have enough storage on your computer to download that kind of data. So this is why it's really important as bioinformaticians to have appropriate data management tools like this. So we can go in and see all of the individual files that we just pulled from SRA. Okay, so that is a wrap on the genome demo and talking about single cell data. At this point, I'd really like to open the floor to any questions that I can answer.
Thank you so much, Nora. It looks like there's a few questions that have been entered into the chat box. And again, feel free to type in if there's more that come up. But the first one is, which methods are used for batch effect correction?
Yeah, so I think I mentioned briefly, we use the Seurat algorithm for batch effect correction. But within Seurat, there's multiple different algorithms that you can use to perform the batch effect correction. So one of those is CCA, which I believe is canonical clustering analysis, and then RPCA, which is reciprocal PCA, or reciprocal principal component analysis, to be more specific. So CCA is great if you have conserved cell types across your samples, and you expect there to be significant shifts in expression based on experimental conditions, or you're taking them from different donors. And then RPCA is better if you may expect to see differences in cell types across your samples. And it's also great if you have really large data sets because it's less computationally intensive.
Great. There's another question on what types of customization can you do in the single cell workflow?
Yeah, so I showed in the Canvas workflow builder the different kind of tools that are running, but essentially anything that you can do in Seurat to customize in the Seurat workflow to customize your analysis, you can do in Genome. So you can adjust filtering parameters, you can adjust the normalization method, you can adjust whether or not you want to regress out technical covariates like UMI counts. All of that is at your disposal. And then we also have the ability to import custom code. So if there is another tool that you want on Genome, we can bring that in for you.
Great. And I think this is a follow-up to that first question around batch correction, asking if there's harmony, and then which version of Seurat are we utilizing?
Yeah, so we use Seurat version 4.3. In a couple of months, we'll be uplifting to Seurat v5, which was recently released. And we don't currently have Harmony available, but as a part of the uplift to Seurat v5, Harmony will be available. And that is through Seurat's tool, Integrate Layers, which also accelerates batch effect correction. So that's nice.
Great. Okay, another question just came in. How can I rerun my single cell analysis with different parameters?
Yeah, so if you have launched a run and you decided that you actually want to go back and rerun your analysis but adjust a few things, you can go into your runs tab and clone a run and then just adjust the parameter. So let's say you're halfway through the analysis, or you've already run the analysis, but you decide, you know what, I want to go back and I want to change the batch effect correction algorithm that I used. I want to change it from RPCA to CCA. So you can clone your run and then just adjust that specific parameter and run again, and it will only run from that point on. So that's time saving, which is really nice because sometimes if you're using, tens of samples, you really do not want to rerun that entire analysis from the beginning.
Okay, a few more. If I don't know what annotation to use, is there one you would recommend I start with?
Yeah, I think a good kind of default is to use the human primary cell atlas because that contains really broad cell types. The only trade-off there is that it doesn't have like fine resolution. So for example, if you're using, like the Monaco data set that I showed, that could show, is it a non-switched memory B cell or a switched memory B cell? With a human primary cell atlas, you're not going to get that level of resolution, but you could see, okay, this is a lung cell or this is a muscle cell, that kind of resolution.
Great. And then one last question, is there a reason why I wouldn't run batch correction? And can I do this with Genome?
Yeah, absolutely. So you do not have to run batch effect correction on Genome. I would say don't. Actually, there's a lot of cases where batch effect correction doesn't occur. Like let's say you did run technical replicates, but they're prepped by the same person on the same day. You probably don't need to do that. And you are expecting to kind of see the same exact thing across samples. So let's say I had 1,000 PBMCs that I prepped at 10:55 and 11 a.m. Like there's probably not going to be significant technical difference there. And then another one is if you have like drastically different things. So you took one sample from the mouse prefrontal cortex and one sample from, I'm not a brain expert, some different part of the brain, you wouldn't expect to see any overlap in those cells. So you would want to process those samples differently and not try to integrate the data there.
Okay. And then another question, what tools are available for downstream analysis?
Yeah, so that's a good question. So for things like tertiary analysis, we do output all of our files. So we produce intermediate files for every step in the workflow. So you can really download the file at any point that you want. And then obviously the output file, the processed file at the end. So you can take that file, it's an R data object, and you could put it into something like cell by gene for further expression, but particularly if you have this R data object, you can definitely import it into R and perform further analysis there if you want.
Perfect. Thank you, Nora. And then I saw a question come in about the recording and when it will be sent. We will send this out after today's webinar, so you should get access to this shortly. And if there are not any other questions, I want to say thank you to Nora, and then have one last screen that I can share just for everyone that is still on the call. We do offer a trial. So if you're interested in testing the software for single cell or public data or RNA-seq, that's something we'd be happy to set up with you. You can either scan the QR code or sign up with the link, and essentially would give you access to Genome. We would work with you, kind of understand your research goals and make sure that we can help from a data analysis standpoint. So thank you everyone so much for joining. Very much so appreciate it. And we will be following up with the recording and reach out with any questions in the meantime. Have a great rest of your day.